Hello students, how are you all? Hope that you all are fine as well as healthy. We are learning biology, chapter number 5, the principles of inheritance and variation. So you know that in last video lecture we have talked about the two main laws of the Mendel, and which are known as the law of dominancy is the first one, and second one is about the law of the segregation. You know that this both different type of laws are dependent upon the result of the monohybrid cross which is done by Mendel for understanding of the inheritance of one gene. Okay, so before starting today's lecture we are going to revise some important aspect, and you know the monohybrid cross means the ratio of monohybrid cross is very much important. So if we write here, we can say that for inheritance of one gene, inheritance of one gene, Mendel did what? We can say that for inheritance of one gene, Mendel did what? Mendel did monohybrid cross. Okay, Mendel did monohybrid cross. So see here, monohybrid cross is the important word. Mono means one. It means in this cross or in this examination or in this practical, at a time only one character will be observed. Okay, we will write here only one character will be observed at the same time, which is known as the monohybrid cross. But the result, okay, means the ratio, means the proportion, means the genotypic ratio and phenotypic ratio of monohybrid cross is very important. So here I am writing the first result, which is known as the genotypic ratio. First result, which is known as the genotypic ratio, and you can say that the genotypic ratio is what? The genotypic ratio is genotypic ratio is 1:2:1. Okay, so you will remember it, 1:2:1 means in F2 generation. See, it is a ratio for or result for which generation? For the F2 generation of the monohybrid cross. In F2 generation, 1 capital T, 2 capital T small t, and 1 small t small t. It means 25% capital T is to 50% capital T small t is to 25% small t small t. It means 1/4 capital T is to 1/2 capital T small t is to 1/4 small t small t. So it's all about the genotypic ratio found in F2 generation during the monohybrid cross done by Mendel. If we talk about the second type of ratio, which is known as the phenotypic ratio. If we talk about the second type of ratio, which is known as what? Which is known as phenotypic ratio, and the result of phenotypic ratio will be 3:1. Okay, the result of phenotypic ratio will be 3:1, means 3 tall and 1 dwarf plant is there. It means 75% tall and 25% dwarf plants are there. It means 3/4 tall and 1/4 dwarf plants are there. So it's all about the result, and with the help of this result we are able to do some examples also. Okay, so then after finishing this question, we had learned about the test cross. You know that the Mendel did test cross, and in test cross with the help of non-phenotype we can get the unknown genotype. Okay, then two different type of laws are there, law of dominancy and law of segregation, and these both laws are known as Mendel's first law of inheritance is equal to law of dominancy, and Mendel's second law of inheritance, which is known as the law of segregation. Law number 1 and law number 2 of inheritance are 100% dependent upon the monohybrid cross. Okay, and today we are going to learn about some different type of question or some exceptions found during the monohybrid cross, and today's question or today's title will be the incomplete dominancy. Okay, it is also most important question. So the title of today's question is what? The title of today's question is incomplete. We can say that incomplete dominancy. The title of today's question is what? The title of today's question is incomplete what? Incomplete dominancy. It is the most important question. Okay, it is a question of 2 marks. It is a question of 2 marks. Okay, see the first point. We can say that if we do the same experiments on other plants as or which Mendel did on pea plant. Okay, what is the scientific name of pea plant? We can say that the scientific name of pea plant will be the Pisum sativum. Scientific name of pea plant will be the Pisum sativum. See, we are doing what? We are doing the same experiments. Okay, we are doing the same monohybrid cross. Okay, which is done by Mendel. Okay, Mendel did this experiment on the pea plant, which is known as Pisum sativum. Okay, then we are getting what? Then we are getting, or we will get different result. We are getting what? Different results. We are getting different results of monohybrid cross. It means if we are doing monohybrid cross, okay, on other plants, means we are not doing the monohybrid cross in Pisum sativum, we are doing monohybrid cross in other plants, then we are getting what? We are getting the different type of result. Means we are not getting this result. Okay, means some exceptions are there. So incomplete dominancy is the example of these exceptions. Okay, so we can say that incomplete dominancy is one type of example of exception. So we can say that incomplete dominancy, it is found in different type of organisms, like first one is dog flower. Okay, which is known as what? Which is known as like snapdragon, which is known as the snapdragon. And second one, okay, the scientific name will be of the gulbas will be the Mirabilis jalapa. You know, Mirabilis jalapa is known as the 4 o'clock plant. It is known as, yes, it is true, but it is known as the 4 o'clock plant. Okay, so we can say that incomplete dominancy is found in different type of plant, like dog flower, which is known as snapdragon, and second one is, see the scientific name of bulbas, it is Mirabilis jalapa, which is known as 4 o'clock plant. Okay, so now if we do the same experiment, means we are doing the monohybrid cross, but we will not take a pea plant, we are taking a different plant. Okay, we are not using the same plant, we are using different plant, but the method will be same. Okay, so if we take the dog flower, point number 3, if we take what? Point number 3, we can say that if we take the dog flower and we are doing what? We are doing the monohybrid cross. We are doing what? We are doing the monohybrid cross. We are doing what? We are doing the monohybrid cross on dog flower. Then you know that according to Mendel, one character having 2 factor. So if we will take the character, and the character is what? Character is about the color of flower. Character is what? Character is about the color of flower. Okay, so this is important. It is character. So if we are talking about the color of flower, flower of what? Flower of dog flower. Then according to Mendel, you know that the one character having the two different type of traits, okay, different type of factor. So the first factor we can say that red color, okay, first one we can say that red color, and second one we can say that whitish or the white colored flower. First one is red and second one is what? Second one is white. If we talk about the sign, then the sign of red color flower will be capital R, and here the sign of white color flower will be capital W. Okay, see, we will in some book or in the in our old syllabus here small r is used, but capital R is not 100% dominant on small r. That's why we are using the W for, okay, W for white, because both are not 100% dominant on each other. Both are incompletely dominant. Okay, that's why see the title is what? Title is the incomplete dominancy. That's why we are using the capital R for red and capital W for white. In our old syllabus of biology, here small r was used, but the result will be same. You are going to take capital R and capital W. Okay, so red color sign will be capital R, white color flower sign will be capital W. And now we are going to do cross, means monohybrid cross. Okay, so here we are taking what? Here we will take true breeder. Okay, we will take here as parent generation, as parent generation we will take here capital R cross with capital W. So this both are what? This both are phenotype or genotype. Okay, think about it. This both are phenotype or genotype. This both are one type of genotype. Okay, and now we are going to write the phenotype. Here the phenotype will be red colored flower, and here we can say that the white colored flower. Okay, here we will write red and white. It is the P generation, means parent generation. Now if we write the gametes, gametes of what? Gametes of P generation. See, these are homozygous or heterozygous. These both are homozygous, and you know that the homozygous will produce the same type of gamete, while heterozygous will produce the different type of gamete in a same proportion, in a same number. So here if we are going to produce the gamete, capital R is the gamete, and here we can say that capital W is the gamete. What about F1 generation? If we talk about the F1 generation, okay, what is the full form of F1 generation? F1 generation means we can say that the filial 1 generation, this both, see here, capital R, capital W. See, it is what? It is a genotype. See, according to Mendel, here in F1 generation, capital T and small t were there. Okay, here capital R and capital W, okay, both are there. And if we talk about the phenotype, then phenotype will be pink in color. See, in F1 generation, we are getting a flower or we are getting a plant which is not there in their parent. Means this plant or this flower is 100% different from their parent. It is the first difference between the monohybrid cross of Mendel and the cross done by the incomplete dominancy. Okay, F1 generation, we are going to follow the same method. So here we will do the self cross. Here we are going to do what? Self cross. Okay, so capital R, capital W is what? Genotype or phenotype? It is one type of genotype. And what about the phenotype? The phenotype will be pink. Okay, so it's all about the F1 generation. Now if you want the F2 generation, then the gametes, okay, you know that the gamete production is important for sexual reproduction. So if we write the gametes of the F1 generation, see here we can say that it is homozygous or heterozygous. These both are heterozygous. So heterozygous will produce the different type of gametes in a different numbers. So we can say that here capital R and capital W gamete is produced, and here we can say that capital R and capital W, this both gametes are produced. Here we can say that 50% capital R and 50% capital W is produced. Here 50% capital R and 50% capital W is produced. So it is all about the gamete production found during the F1 generation. Now if we write here the F2 generation, okay, everyone see here, if we write here the F2 generation, and here we are going to use the Punnett square. So write a one sentence using Punnett square. So using Punnett square, okay, so you know that with the help of Punnett square we are able to get the possible genotypes. Okay, we are writing here gametes capital R, here capital W, suppose it is a male gamete, here female gamete, capital R, capital W. Okay, see here the sign are important. Okay, now see we can say that here we are getting what? Here we are getting capital R, capital R, capital R, capital W, capital R, capital W, capital W and capital W. These all four are what? These all four are one type of what? One type of genotype. See why we are getting only 4 genotype? Because you know that the number of male gamete multiplied by number of female gamete. Okay, it is a one equation. If you want to know the number of possible genotypes, then number of possible genotypes is equal to number of male gamete multiplied by number of what? Number of female gamete. So 2 multiplied by 2 is equal to what? Is equal to 4. So here we can say that if we write the phenotype, it will be the red. Yes or no? Here RW means pink, RW means pink, WW means what? Means white. So it's all about the F2 generation. Now if we write the main thing, okay, now if we are going to write the result, then first result is what? First result is about the genotypic ratio. The first result is about the genotypic ratio. Then see here we can say that 1 RR is to 2 RW is to 1 WW. Okay, so we can say that the genotypic ratio is what? The genotypic ratio is 1 is to 2 is to 1. The genotypic ratio is the 1 is to 2 is to 1. Okay, so see we can say that genotypic ratio of both will be same. According to Mendel's monohybrid cross, it is 1 is to 2 is to 1. According to incomplete dominance, it is 1 is to 2 is to 1. But if we talk about the second option, which is known as the phenotypic ratio, if we talk about the second option, which is known as the phenotypic ratio, then phenotypic ratio 1 red colored flower is there, 2 pink color flowered is there, and 1 white color flowered is there. It means it will be 1 is to 2 is to 1. See here, according to Mendel's monohybrid cross, according to it the result will be 3 is to 1, and here we are getting phenotypic ratio 1 is to 2 is to 1. It means we can say that in incomplete dominancy the genotypic ratio and phenotypic ratio both are same. See, both are 1 is to 2 is to 1. Okay, so it's all about the incomplete dominancy. So total main 2 differences are there between incomplete dominancy and the monohybrid cross done by 1. First difference is what? First one is we are getting the different phenotype of a plant, okay, which is not there in parent, and second one is phenotypic ratio of both are different. Okay, so the part number 1 of today's question is completed. See, this question is 100% important. These names are important. These all are one type of MCQ. So part number 1 of today's question is completed, and now we will talk about some different type of very short concept. Okay, first of all we will talk about the concept of the dominancy. Okay, see here, there is a short paragraph in your textbook of the concept of dominancy. Okay, see here we can say that if we talk about the understanding of what? Understanding of concept of dominancy. Understanding of what? Understanding of concept of what? The concept of dominancy. Okay, see here we can say that first point. Okay, what is dominancy? It is the first question. What is dominancy? Second question, why some factors are dominant and why other factors are recessive. Okay, why dominant and recessive, both different type of factors are there. Okay, so these are the some basic questions, but we can say that if we write some points about the gene, see gene, if we talk about the gene, then gene, it is very important. Gene is very important because we can say that because the characters are expressed. The characters are expressed because of genes. See, this sentence is very much important. The expression of characters, is 100% controlled by what? Is 100% controlled by gene. The expression of what? Expression of character means genes are the what? Means we can say that genes are the responsible for the expression of characters. Okay, you know that we all are humans, we having the special type of characters because of what? Because of the special type of genes. Okay, so the genes of human and genes of ant, both are not same, both are different. Okay, but it is true that both are one type of animal, but our genes are not same. Even if we talk about the you and me both are human, but the gene of you and me will not be same, it will be different. Okay, so genes are responsible for the different type of characters. So expression of characters. So for characters, expression is there. Now see, point number 2, we can say that we humans are one type of what? We humans are one type of diploid organisms. We humans are one type of diploid organisms. Okay, see, what is the meaning of diploid organisms? Diploid organisms means we can say that we all are of 2n type. 2n means where n is equal to what? When n is equal to number of chromosomes. Yes or no? Where n is equal to what? N is equal to number of chromosomes. So we all humans are one type of diploid organisms. It means 2n. How 2n is formed? We can say that n, okay, from the mother means half number of chromosomes we are getting from the mother and half number of chromosomes we are getting from the father. Okay, so maternal and paternal, 50% maternal, 50% paternal, means we can say that 23 number of chromosome from mother and 23 from the father, that's 46 chromosomes are there. That's how total 46 chromosomes are there. Yes or no? So we all humans are diploid organism. It means we are getting a pair of gene from mother and a pair of gene from a father. Means if we talk about a one pair, see, it is a one pair of gene, then this allele or this factor or this member will come from mother and that member will come from the father. That's why humans are always heterozygous. We all are not homozygous. Okay, you know that the dominancy is only applicable in the heterozygous condition. If condition is homozygous, then dominancy is not required. But see, in humans always heterozygous condition is there because one member will come from the maternal and one from the paternal. So we can say that point number 3, in diploid type of organism, we can say that gene pair will be always heterozygous. Gene pair will be always heterozygous, and you know that in heterozygous, one is what? One is, we can say that dominant, and what about the other one? Other means second will be always the recessive. Okay, so it is what? It is a 100% total understanding of the concept of dominancy. We all are diploid means we having the heterozygous condition. Why heterozygous condition? Means one member, one gene or one allele is coming from the mother and one from the father. Okay, that's why we are heterozygous and the rule of dominance is applicable. Okay, so two different type of concepts are completed. In one concept we have learned about the incomplete dominance, in second concept we have talked about the understanding of concept of gene. Okay, so today's lecture is 100% completed. I hope that you are getting it very easily. Thank you very much.